Hello, welcome student. We shall study today the structure of protein. It is also a topic of biomolecules. Now the structure of protein, we know that protein is a polymer, and in the polymer it is a the heteropolymer, means different types of molecules attached to it. And second, the combination of amino acids, and these amino acids are combined with one another by means of peptide bond. So it is called polypeptides. So these are the protein. These are features of the protein. Now the structure of protein, structurally it is classified into primary protein, secondary protein, tertiary protein, quaternary protein. That is primary, secondary, tertiary and quaternary. It is also known as 1 degree protein, 2 degree protein, these are also known as 1 degree, 2 degree, 3 degree and 1 degree, like that. Now first we shall see what is the primary protein. In the primary proteins, the amino acids link to each other by means of peptide bond. They are combined to each other, that is amino acids A1, A2, A3 and so on. Understand? So this is they attached each other by means of peptide bonds. And I have already told you that peptide bond is CONH. This bond between two amino acids, here amino acid 1, here is amino acid 2, amino acid 1 and amino acid 2. These are combined with each other by means of CONH bond, that is called peptide bond. I have already discussed about this in the previous video. So these bonds are found so many. As many the amino acids are found, there is one less peptide bond. जितने भी amino acids होंगे उसमें उससे एक कम peptide bond होगा. Understand? That is, now suppose it is the first amino acid and this is the last amino acid. the first amino acid will be called N-terminal and the last amino acid is called C-terminal. So in such a, by means of peptide bond, it is in a chain. It will be in a chain in a linear position, and in which many many amino acids will attach to each other by means of पेप्टाइड बॉन्ड, जिसमें बहुत से अमीनो एसिड एक लाइन में एक चेन में एक दूसरे से पेप्टाइड बॉन्ड से मिले होंगे. सच टाइप ऑफ कंडीशन इज नोन एज प्राइमरी प्रोटीन. अंडरस्टैंड? नेक्स्ट वन इज सेकेंडरी प्रोटीन, in which amino acids are found in a coiling forms. When they increase, they become coil. They are found in form of a coil. They are found in the form of a coil. And second, these are the amino acids. These are the amino acids are found on the chains. These amino acids are attached with each other by means of peptide bond. By means of peptide bonds. So these are peptide bonds. These are peptide bonds. Now, in the coiling, where two amino acids are just in front of each other. यहाँ देखो कॉइलिंग में दो एमिनो एसिड एक दूसरे के सामने है. तो इन एडिशन टू पेप्टाइड बॉन्ड देयर इज हाइड्रोजन बॉन्ड इज आल्सो फॉर्म्ड. In addition to, and due to this hydrogen bonds, it becomes coil. तो इसमें हाइड्रोजन बॉन्ड होने की वजह से ही क्या होता है, हाइड्रोजन बॉन्ड मीन्स व्हेन हाइड्रोजन इज फॉर्म एन एक्स्ट्रा बॉन्ड विथ हाईली इलेक्ट्रोनेगेटिव एलिमेंट सच एज ON etc. जब हाइड्रोजन सामान्य बंधन से हटकर एक अलग बंधन, वो भी किसके साथ, इलेक्ट्रोनेगेटिव एलिमेंट्स जैसे नाइट्रोजन ऑक्सीजन, इन सबों के साथ जब वो एक अलग बंधन बनाता है तो वो क्या होता है, हाइड्रोजन बॉन्ड कहलाता है. अब हाइड्रोजन बॉन्ड दो तरह के होते हैं, इंटर हाइड्रोजन बॉन्ड एंड इंट्रा हाइड्रोजन बॉन्ड. जब एक ही मॉलिक्यूल्स के दो मतलब दो एलिमेंट्स के बीच में बनता हो तो वह इंटर, और जब अलग अलग दो हो तो वो इंट्रा. यानी एक इंटर मीन्स, यानी सपोज समझ लो ये एक है, यहाँ एलिमेंट्स है और यहाँ एक एलिमेंट्स है, तो इन दोनों के बीच में अगर हाइड्रोजन बनता है तो बिटवीन दैट इज इंटर हाइड्रोजन बॉन्ड. और जब मान लो एक ही है और एक ही के बीच में बनता है तो दैट इज इंट्रा. इंट्रा मीन्स विद इन. तो जब एक ही में बने, समझे, तो वो इंट्रा होगा. और जब दो अलग अलग स्ट्रैंड के बीच में एमिनो एसिड बने, मतलब हाइड्रोजन बॉन्ड बने, तो उसको कहते हैं इंटर. इसलिए इंटर मतलब बिटवीन. तो बिटवीन जहां भी होगा तो वहां दो के बीच होगा, और इंटर मतलब विद इन, उसी में, यानी एक ही के अंदर. अब जैसे यहाँ जो है एक ही में बना है, तो दैट इज इंटर. तो यहाँ क्या होता है, तो ये कॉइलिंग किसके वजह से बनता है, ड्यू टू हाइड्रोजन बॉन्ड. तो इसमें क्या होता है, ये कॉइलिंग स्ट्रक्चर्स है, and in addition to hydrogen peptide bond, the hydrogen bond is also found, so it becomes a coil like structure. Now the third degree, that is tertiary bond. Now we shall discuss the tertiary protein. You know that sometimes that is a coiling, but it will increase and forms in such like type of structures, it becomes a coiled like structures, and it becomes a so much increase, that is coiling, and it also become a hollow like structures. And so there are amino acids. So the amino acids are found. So in between that, how many bonds are formed? One is, first bond is a structure is hollow like peptide bond. Understand? Second, hydrogen bond also formed. And next one, due to electrostatic force, the ionic bond is also formed. Ionic bond, that is due to electrostatic force of attraction. This is due to electrostatic force of attraction. The ionic bonds are also formed. Means that is transfer bonds, which is formed by the transfer of electrons from one atom to another. That is the ionic bond. And next one, if there will be sulfur containing amino acids, यदि इसमें सल्फर containing amino acid, जैसे सल्फर containing amino acid जैसे cysteine, methionine, etc. अगर ये जब अमीनो एसिड होंगे तो उनके सल्फर के बीच में भी बॉन्ड्स बनेगा, दैट इज नोन एज डाई सल्फाइड बॉन्ड्स। तो ये इतने टाइप के बॉन्ड्स इसमें पाए जाएंगे, समझे। तो टर्शियरी स्ट्रक्चर्स में क्या हुआ कि ये जो कॉइलिंग होते हुए कॉइलिंग जो इंक्रीज करते जाएगा, एंड दैट बिकम्स ए हॉलो लाइक स्ट्रक्चर्स। ये जो इसमें हम यू विल सी द पिक्चर इन योर बुक। तो सो सच ए हॉलो लाइक स्ट्रक्चर्स इज फॉर्म्ड in tertiary protein. We may also write in. So we can increase in that. That is hollow, and in that protein, such proteins, these 4 types of bonds in these proteins, tertiary protein, these types of bonds are found. And if the sulfur is present, sulfur containing amino acids is present, that will contain sulfide bond. Now the quaternary protein. This protein is mixture of some protein. In some there are in such type of protein is a complex protein, a cube-like protein. Complex cube-like protein is formed. Understand? And that is found in the hemoglobin like that. So this such type of protein is found when so many proteins are combined to each other in such a fashion. They intersect to one another, and that protein is called quaternary protein. And in that the amino acids are found, and in that protein all the type means peptide bond, hydrogen bond, ionic bond, and disulfide bond. All the bonds are formed, and in the hydrogen both inter and inter. Both type of hydrogen, inter means within the same and between different, that is inter, and in the same, that is inter. जब एक ही में होगा, मान लो यहाँ से यहाँ बना तो ये हो गया इंटर, और जब इसमें से इसमें बना, ये दोनों दो अलग अलग स्ट्रैंड्स हैं, इन दोनों में बना तो इंटर हाइड्रोजन बॉन्ड, तो दोनों इसमें इंटर हाइड्रोजन बॉन्ड्स दोनों पाए जाते हैं। और यदि उसमें सल्फर कंटेनिंग अमीनो एसिड्स हुआ तो सल्फाइड बॉन्ड यानी डाइसल्फाइड बॉन्ड्स भी पाए जाएंगे। एंड इट्स स्ट्रक्चर इज कॉम्प्लेक्स cube like. So all such type of bonds are found which makes them complex. They in the some proteins are coiling proteins are intersect to each other. Now we shall discuss nature of bond linking monomers in a polymer. You know, we have started there about polysaccharide, that is a carbohydrate, protein, next nucleic acid, and the about that. Now the polysaccharides, we know that it is the combination of monosaccharides. There are monomany-saccharides, that is called, and protein is the combination of amino acids. It is the combination of amino acids. And nucleic acid is the combination of nucleotides. Understand? These are the combination. And lipid is the acid of fatty acid plus glycerol, that we already we have discussed. So these are the monosaccharides. That is polysaccharide is the combination of monosaccharides. Protein is the combination of amino acids, and nucleic acid is the combination of nucleotides. In the polysaccharides, we have studied that one example is starch. It is the polyglucose. Because same type of molecules take part in the formation of starch, so it is the non-H. Homopolymer, and means only glucose are combined. And we have already told you that one, two, three, 4, 5, 6 CH2OH. In the same way, this is the glucose, understand? This is the glucose molecules, understand? And in the glucose molecules, I have already in the last video that these two molecules are, suppose hydrogen the molecules. Yeah, combined with each other by means of glycosidic bond. So two monomers, or in that way the glucose molecules are, many glucose molecules are attached to each other by means of glycosidic bond, and so a big polysaccharides is formed. इसमें बहुत से ग्लूकोज मॉलिक्यूल्स एक दूसरे से ग्लाइकोसिडिक बॉन्ड के माध्यम से मिलते जाते हैं, और जितना ग्लाइकोसिडिक बॉन्ड बनता है उतना हाइड्रोजन वाटर मॉलिक्यूल्स मुक्त होता है। So in this way, many units of monosaccharides combines to each other and forms the polysaccharide altogether. And about protein, we have already studied protein, amino acids 1, amino 2, they are attached to each other by means of peptide bond. I have already told you that 2 amino acids, suppose this. This is amino acid 1, this is amino acid 2. When they will combine to each other, that H and OH will mix to each other and they formed a bond that is known as peptide bond प्लस H2। जो H और OH आपस में मिले थे वो H2 बन के निकल गया और यहाँ एक बॉन्ड बन गया, दैट इज पेप्टाइड बॉन्ड। इस तरह से जो भी अमीनो एसिड आपस में मिलेंगे एक दूसरे से, दैट विल so as much the amino peptide bond will forms, these other amino acids will attach to each other and forms a protein। मतलब जितने अमीनो एसिड आपस में मिलेंगे समझे, इस तरह वो आपस में मिलके पेप्टाइड बॉन्ड के माध्यम से एक लंबा चेन बना लेंगे एंड दैट विल बी ए प्रोटीन। एंड द फर्स्ट प्रोटीन इज कॉल्ड एन टर्मिनल्स एंड द लास्ट अमीनो एसिड इज कॉल्ड सी। यानी पहले अमीनो एसिड को एन टर्मिनल्स कहते हैं और लास्ट वाले अमीनो एसिड को सी टर्मिनल्स कहते हैं। और बीच में जितने भी ये होंगे अमीनो एसिड होंगे सब एक दूसरे से पेप्टाइड बॉन्ड के माध्यम से लगे हुए होंगे।